So it's a pleasure to welcome Natalia Komarova. She's apparently the Chancellor's Professor of Mathematics at UC Irvine. And, you know, Natalia is really a veteran of the mathematical oncology field. She's got interests in not only mathematical modeling of cancer, but also sort of complex social phenomena as well, and more generally kind of evolutionary processes. So she got her PhD in '98, and that was with AC Newell. And where was that, Natalia?
Arizona.
Arizona. And then got a research assistant professor in applied maths at Arizona, and then moved to University of Warwick as a research fellow, I guess a short stint there for a year, and then moved to University of Chicago as a visiting scholar, and then was at the Institute for Advanced Study for a whole 5 years. And I guess that's where you did some work with Martin Novak. And then became assistant professor in mathematics at Rutgers in 2003, and then eventually landed in UC Irvine in 2004. And so Natalia has been doing lots of very cool work, both independent of her husband, but also with her husband. And of course, they've written a rather well-read book that many of us know about, and it's a great resource. And I guess they published an update to that not that long ago. Is that right, Natalia?
Yeah, yes. It's really a new book.
It's a new book. There you go. So it's a pleasure to have her here, and we're really looking forward to hearing a somewhat general title of Mathematical Modeling Cancer Evolution. So take it away, Natalia.
Thank you so much for this invitation. It's a pleasure to speak here, and I've never traveled to Moffitt. So now with this new life, it makes travel easy. So I'm here. And it's my pleasure to talk to you about some of my work on cancer. So I will structure it in the following way. I'll talk about two types of mutations that are important in cancer evolution, which we call loss of function and gain of function mutations. So part one will be about loss of function, which is another way to say tumor suppressor genes. And I'll illustrate this with some evolutionary findings and talk about passenger mutations and their possible role in cancer. And then we'll move on to gain of function mutations, for example, oncogenes or any other one event mutants. And we'll look at mutants in expanding colonies and evolution in random environments. So the progression of colon cancer illustrates the two types of mutations that I want to talk about it. The first, one of the early events is an activation of a tumor suppressor gene APC, and this is a loss of function mutation. And then we have a typical event, which is an activation of a KRAS oncogene, and this is an example of a gain of function mutation. So what's the difference? Gain of function is a one mutation event, right? You have a gene and something changes, there's a genetic change and a function turns on. So the properties of the new cell are different from the wild type cell, for example, increased fitness. Loss of function takes two steps. There are two copies of a tumor suppressor gene. You inactivate one and nothing much happens, or even the fitness of this intermediate cell is lower than the wild type. And then you inactivate the second copy of the tumor suppressor gene, and then the fitness grows because this gene that exists in two copies keeps the cell from doing bad things, from growing too fast, from dying too little. So basically you have to inactivate both copies to see this super mutant that can invade. So this is basically a theoretical kind of framework. And loss of function, a two-step process, is a very slow evolutionary step, right? It may get worse before it gets better. So this is what we call crossing of a fitness valley. So a wild type cell has fitness, say relative fitness 1, let's call it 1, then it can get worse when you inactivate the first copy, and then it gets better. Or you can have a fitness plateau or even fitness foothill like this. So we want to know what is the probability that by time t, both mutants have been inactivated. So here I illustrated with an example of bacterium flagella. This is just a general evolutionary example. So if you have a bacterium, at some point it evolved a flagellum that helps it swim. Okay, so basically it probably was okay before the flagellum was in place, and it became better after the flagellum evolved, but there was an intermediate stage where it had the flagellum, but it wasn't a very good one. So this is kind of an illustration of a fitness valley. So you start here, then you have to, it has to get worse before it gets better, and that can take a long evolutionary time. So in cancer, this happens all the time. This is a rate limiting step in carcinogenesis. And I'll tell you about some ways in which we can handle this process in different settings, starting from the simplest one. So here is a model that is very primitive, but we have to start somewhere. So this is what they call a Moran process. So you have a bunch of cells, a constant number of cells in a box. You kill one and you reproduce another one, such that each event is a pair of events and a birth and a death. And that you just keep going and you watch the evolution of a mutant. So when I was still a postdoc, we discovered the phenomenon of stochastic tunneling, where you start with a whole compartment filled with wild-type cells. And then instead of kind of fixating into this intermediate state, you tunnel through and evolve the super mutant without having stopped, making this intermediate stop at the intermediate phenotype. So, and there are mathematical methods of calculating this rate of tunneling, which is a good way to characterize the rate of evolution. So it is possible to calculate this rate under different circumstances. So here, this tunneling rate is calculated for this primitive mass action model. And it's a function of the compartment size, like basically how many cells are in the box, and the first and the second mutation rate. And it depends on whether the intermediate mutants is neutral or advantageous. So for our purposes, it's just important to know that we know how to calculate it, right? So now we move on to something a little bit more realistic, although here it doesn't look realistic. So we look at the one-dimensional spatial system. Okay, and now it goes like this. You kill a cell and you reproduce another cell, but this cell has to be a neighbor, right? So we keep track of the spatial structure of the population. In this case, it's 1D. And in this case, we can calculate this rate, and it's faster. Okay, as soon as you add spatial restriction, evolution happens faster. And of course, you can do it in 2D and 3D, right? Just in the same way, you kill a cell and you only allow a neighbor to reproduce and replace it. And you get exactly the same result. If you compare the rate of evolution in this more realistic, well, presumably more realistic two-dimensional or three-dimensional spatial system, it happens faster than in a system that is well mixed, or what we call a mass action. And here it's easy to explain why this happens. So here is an example of a simulation where red represents wild-type cells and blue represents intermediate mutants. Intermediate mutants pop up here and there, and this is a non-spatial mass action system, so they can appear anywhere. And when they reproduce, they place their offspring anywhere, right? Because it's well mixed. And then they have to compete with their neighbors. So if the mutants are somehow weaker than the wild-type cells, they have to compete with everybody, and they normally lose. In a spatial system, mutants tend to stick to these mutant islands because, you know, when they reproduce, they place their offspring next to themselves. And now they have an easier life because instead of competing with everybody, they only compete with their neighbors, and their neighbors are as weak as they are. so they stick around for longer. And so there are more of them at each moment of time. There's the probability to have such an island is higher than here. And they produce the super mutant faster. So that's why evolution progresses faster in the spatial system. Now, we want to add a third component, hierarchy, cellular hierarchy. As we know, in many cancers, hierarchical structure and lineage structure is important. So in this very simple model, we have a single stem cell, which gives rise to a lineage of cells. We get rid of the differentiated cells, and then we consecutively divide the other cells until the stem cell divides and replenishes this whole thing. And this process proceeds, right? So we can calculate the rate of evolution here too, and it turns out that it's slower. Okay, so somehow we have this hierarchy of rates. So in the middle, and this is a mutation rate, and this is the rate of evolution. In the middle, we have this simple primitive mass action system. And in any spatial model, 1D, 2D, 3D, evolution happens faster. In a hierarchical model, evolution happens slower. So we can actually dig in more into hierarchical systems. The one I showed you before is very primitive. It's a very rigid system where we have this perfect tree of doublings, right? It's not like this in reality. And so a step toward reality would be to look at different types of cell divisions. So here, a stem cell can divide in 3 different ways, right? It can divide into 2 stem cells. It can divide and differentiate into 2 daughter cells, or it can divide asymmetrically into a stem cell and a daughter cell. And a differentiated cell here, we assume that divides, right? So all these things lead to a decision tree that cells have to make in order to turn over and maintain homeostasis. So stem cell divides, it has to decide whether it divides symmetrically or asymmetrically. So asymmetric goes like this. If it's symmetric, it needs to decide whether it's going to proliferate or self-renew, that is give rise to 2 stem cells, or it can differentiate and give rise to 2 differentiated cells. So it's quite complicated, right? But nonetheless, it's possible to analytically calculate the rate of evolution here too. And now this model is better because it actually distinguishes between, for example, symmetric and asymmetric divisions of stem cells. And interestingly, we found that the rate of evolution is sensitive toward what kind of decisions the stem cells make. They can decide to always divide symmetrically or always divide asymmetrically or something in between, right? So it turns out that the rate of evolution is minimized by symmetric divisions. And the difference here can be huge, depending on some parameters. It could be like a 1,000-fold difference, right? Or 10,000-fold difference between always dividing symmetrically or always dividing asymmetrically. Basically, there are different models. For example, in connection with colonic crypts, there has been literature. First, people used to believe that stem cells divided always asymmetrically, and that was kind of like a trademark of stemness, right? You divide and you give rise to 2 different types of offspring, stem cell and the more differentiated daughter cell. But recently, at least in the context of colon, people were saying that it's really more symmetric divisions. It's a balance between between sometimes self-renewing, so giving rise to 2 stem cells, and other times differentiating, that is giving rise to 2 differentiated cells. So this is a better process from the evolutionary point of view because it slows down the rate at which double-hit mutants are produced. So it delays the onset of a cancer, right? So, so what we have seen so far is that 2-hit mutant creation is the slowest in hierarchical tissues compared to homogeneous tissues. And it's absolutely the slowest for symmetrically dividing stem cell lineages. So this could be one of the reasons why evolution chose a hierarchical organization of organ tissues, especially epithelial tissues. This is to delay the onset of cancer, to push it beyond the reproductive rate, and to prolong the lifespan of the species. So I will give you some other application of this type of theories, which has to do with the types of mutants that are thought to be involved in carcinogenesis. So as you know, there are two types of mutations that people talk about. There are driver mutations, which actually confer selective advantage to cells and enable them to break out of homeostasis. So these are oncogenes and tumor suppressor genes, right? Or genes responsible for genetic instability. And they're passenger mutations. They are just random mutations that create useless cells. They are usually deleterious. They do not contribute directly to the disease, right? So, and normally the focus is on these, right? These are unimportant. So here I show you that the passenger mutations can be important. So let's again consider the process of inactivation of a tumor suppressor gene. So here both copies are intact. Here one of the copies is inactivated, and here both copies are inactivated. And now we add, so, and this is the process of creating a driver mutation, right? And here is everything else. The passenger mutations, they are denoted by crosses, right? So you can go from here to here and create all these weird cells that most of them are lower fitness or even deleterious. There's a whole host of these mutations. So we put them all together in this one pot. And such damaged cells can also go down the pathway of inactivating the tumor suppressor gene. And it turns out that if you make the calculations, this pathway to cancer, to the first step in cancer, is faster. So basically, instead of going straight, it's somehow easier to first get a bunch of deleterious mutations, to passenger mutations, and then go down this pathway. So it's interesting, and you can actually understand it intuitively. So the passenger mutations lower the overall fitness of the cell population. So, and to go from here to here, as I illustrated, you have to go down a fitness valley. So it has to get worse before it gets better. That takes a long time. So when you lower down the overall fitness, your valley that you have to cross becomes shallower. So actually you gain and you cross it faster. So there is a speed up of evolution if you go that way. And it's easier to cross the evolutionary valley. This is a calculation that shows you how much the presence of passenger mutations accelerates evolution compared to the absence of passenger mutations, right? You can calculate it. So it's not possible in reality to compare the presence of passenger mutations with the absence, because they're always there, right? We could not design an experiment where there are no passenger mutations, but we can evaluate their effect by comparing them with another known phenomenon, which is the Lynch syndrome. As you probably know, Lynch syndrome is a familiar disease where the patients have a germline mutation in one of the mismatch repair genes. So these cells that they have, the cells that they have one inactivated copy of in that mismatch repair. When they acquire the second copy, these cells mutate faster, basically. So this mutation rate is faster than normal. This mutation rate is fast. So if you first acquire that second mutation that makes you a mutator, you go down faster. Okay, so it accelerates the road to cancer, and normally these there's an earlier onset and a higher probability to develop colon cancer and other cancers. So what is the size of this effect, right? So we can evaluate the effect of passenger mutations by comparing them with the accelerating role of the Lynch syndrome. And depending on parameters, it is comparable with that. So the fold acceleration in the rate of evolution that is given by the presence of passenger mutations is within the range of possible acceleration that comes from the Lynch syndrome. So basically the message here is that passenger mutations are important and should not be ignored, because they create an environment that changes things and can even kick the evolution to proceed faster. So in the second part of my talk, I'm going to switch gears and talk about something that is actually conceptually simpler. It's a one-step process, gain of function mutations. So it's only one event. And we'll talk about both advantages, disadvantages, and neutral mutations. And I will start with a very new work that we did this year, mutant evolution in spatially structured populations. So this is a two-dimensional simulation where blue are wild type cells and yellow are mutants. And you start with a small seed in the colony and you let it expand and cells can mutate and you develop these interesting patterns, right? And we will be comparing again a mass action system where cells can divide and place their offspring anywhere, and spatial systems that grow in a one-dimensional, two-dimensional, three-dimensional space, right? So, yes?
Quick question. Is there death in this model?
In this one?
Yeah.
Yes, we'll be in a second.
Okay.
So this picture is without death, and just wait for a few seconds, okay? So for this work, in the absence of death, was done in this paper. And this is the type of patterns that were obtained. And there are two types of colonies, the shapes, right? They're called wedges and bubbles, right? So we start with one cell in the center, run the spatial model until the total number of cells reaches n, count the number of mutants, repeat this with the mass action model, and do we expect more or fewer mutants in the spatial colony compared to the mass action? So it turns out, according to this very important paper, that in space, in this model, you always get more mutants. And the explanation is so-called jackpot events, where mutants that happen early on give rise to large colonies. So this is how it works. If you have a mass action or well-mixed model, jackpot mutations are very rare. So this is, if you start with one cell, then there are 2, 4, 8, 16. If you mutate here, then this gives rise to a huge colony. But the probability of that is extremely low because it has to happen in the beginning. If a mutation happens at the end, and that's when most mutations happen, it doesn't really make such a big difference. So in an exponentially expanding colony, jackpot mutations are very rare. If you have a spatial model, so this is an extreme spatial model, 1D, you start here, you divide, you grow this thing, but once you mutate, it's enough to mutate once. Everybody else behind you is going to be a mutant. So this is an extreme mathematical abstraction, right, of space, one-dimensional space. And here a jackpot mutation can happen anytime, right? It can happen here, here, here. It still gives rise to a huge colony. So jackpot mutants are much more important in space. And so it works the same way in 2D, 3D, but this is just the most extreme scenario. Indeed, it was obtained that there are more mutants in space, and these are simulations. These are our simulations, but they were performed by other people as well. So this is a neutral mutants. When you have advantageous mutants, you mostly have wedges, and when you have disadvantageous mutants, you have bubbles because they get closed up like this. So we add death, and we see something similar. This is neutral, both wedges and bubbles, and this is advantageous and disadvantageous. We performed simulations to confirm that neutral mutants there are more of them in space. So this is the function of a death rate, mass action space, more mutants in space. So we basically fix the colony size at n and compare the number, statistically, the number of mutants in the spatial and non-spatial colony. Advantageous mutants, you can be advantageous by increased divisions or decreased death, always more mutants in space. Disadvantageous, decreased divisions, more mutants in space. But here is a difference. If you are disadvantageous because you die more, then we have the opposite result, actually. Starting from some threshold disadvantage, there are more mutants in mass action than in space. So it's the opposite result of everything else. This effect actually becomes bigger as the colony size increases. So it takes a lower threshold. So and here is a possible explanation of this that we came up with. So, you know, when you don't understand something, you look at ODEs. So we wrote down ODEs with wild type of mutant cells. This is just logistic growth, production by mutations from X to Y, and death. And we wanted to kind of emulate the effect of space by running Gillespie simulations in an island model, right? Metapopulation model. So we have X, I, Y, I. They are the number of mutants and wild type cells in island number I. So islands communicate with each other by migration here. And so it's a stochastic system because we use Gillespie simulations, and we can watch the appearance of mutants in this system. And so these are the results for this metapopulation or island model. So these coordinate systems are like this. This is the division rate of wild type. I take it 1. And this is the death rate of wild type. So I fix the properties of the wild type cells. And I'm going to take mutants as points in this space. So these points are characterized by mutant division rate that is smaller than 1. These mutants have a decreased division rate. And these mutants have the same division rate as the wild type, but they have an increased death rate. So you can, there are 2 different ways to give a cell a disadvantage, right? So we fixed the parameters of the wild type cells, picked different parameters for disadvantageous mutants, and explore what happens, right? So we'll be comparing a metapopulation model, which emulates spatial relationships, and the mass action model, which is just ODEs simulated by Gillespie. So first we look at these decreased divisions. So here the division rate of mutants is smaller. And we can see that for these points, 1, 2, 3, 4, 1, 2, 3, 4, there are fewer mutants in mass action and more mutants in the spatial model, as expected. Next, we're looking at these points that have increased death. And you can see that if the disadvantage is large enough, there are actually fewer mutants in space. So the results for this very primitive simulation, which is a metapopulation patch model emulation of spatial relationship, it's the same result as in true spatial simulation, right? Okay.
Hi, this is Heiko. Can I ask a quick question, please?
Of course.
So in your mean number of mutants, you show us the average, but both of these simulations are stochastic. Could you comment on the error bars for those?
Yes. So I don't think I have a slide with we did the full distributions actually, and the distributions are pretty wide, but so if you plot standard error here as a simple measure of statistical significance, you absolutely get a statistically significant result. And if you look at the distributions, they are skewed in a different way, right? So in mass action and metapopulation, these results are presented in this paper. So yes, this is a statistically significant result, despite the fact that the distribution is pretty wide, as always in such simulations.
Do you know how the distributions are skewed? In what directions are they skewed?
Well, so if you plot the logs, well, I know this general shape, right? I remember from the paper, but so there are longer tails. So here with these ones, there are longer tails for metapopulations. And here there are longer tails for mass action. That's what happens.
Okay, thank you so much.
Okay. Here's an interesting result. So here we say, okay, the metapopulation model emulates space because we allow migration between neighboring demes, between neighboring islands. So it's like space, right? You go to your neighboring things. And now we look at the metapopulation model like this. Basically, you can migrate anywhere you like. So it's not really spatial. It's fragmented, but not spatial. And amazingly, you have exactly the same result, almost no difference. I didn't even plot a different picture. So spatial, truly spatial island model and a non-spatial but fragmented model give rise to the same results. So how do we explain it? So here's an explanation. Again, we'll go back to ODEs, like really ODEs just in one patch. Let's plot the number of mutants as a function of time. And we will do it twice. This is for mutants that have a decreased division rate, and this is for mutants that have an increased death rate. So you know that when we have a logistic growth model, mutants that are produced will reach a selection mutation balance. But these two types of disadvantageous mutants reach this selection mutation balance differently. The ones that have decreased divisions grow toward it. And the ones with increased death overshoot and come down to it. Because at the beginning, they don't know that they are disadvantageous. They just divide. They have the same division rate as everybody else. And then death is a smaller rate than division. So it takes a bit of time to realize that you're disadvantageous. So you come down to it. So it goes like this. So now let's plot the number of mutants as a function of the total population size. And you see that for these two types of mutants, this has a different concavity. It goes up and down or down and up. So for increased death, the mutants are maximized before, so the horizontal axis is the population size. When the colony is full, it doesn't maximize the number of mutants. They're maximized before that. But for decreased divisions, the number of mutants is maximized when the colony is full. Okay, so here's a comparison between a fragmented model and a mass action model. Mass action model can be represented as a bunch of these demes that are identical to each other and they all move in sync. Right? That's so you take that ODE and you just make a bunch of identical ODEs and add them together. This is mass action model. And this is a true fragmented model. In a fragmented model, if you put your mutant here, it spreads through the demes. So the spread is desynchronized in different demes. So let's measure the number of mutants at a certain colony size. Here in mass action, it will correspond to all the demes being kind of half full. In the fragmented model, it will correspond to some demes being completely full and others completely empty and a couple in between, right? So this is the difference. And now you can see that for those mutants that are maximized by full colony, you will have more of them. So here, for decreased divisions, maximum percentage of mutants corresponds to the patches that are completely full. So we have more mutants in the fragmented systems. And for increased death, these ones, we are actually maximized at an intermediate size. So mass action will have more mutants. So the explanation here is not jackpot mutations, but something else, this phenomenon of desynchronization that happens in the fragmented system. So the next bit with regards to mutant growth in space is we looked at different geometries and we wanted to derive growth laws. And in the literature, there were a few of those derived before, but we came up with a simple method, right? And we could do it systematically for any geometry. So we looked at a two-dimensional, like a rectangle growing in one side, like this 2D flat front. 2D range, we have a circle expanding in all directions in 2D. Then we have a cylinder.
Yeah.
Yeah.
Can I ask before you go on to this? So your conclusions there seem to imply that the death rate didn't have a huge impact overall.
Say again, what didn't have a...
The death rate.
Well, I wouldn't say that. So I guess the conclusion here is that in the case of disadvantageous mutants, it's very important in which way the disadvantage is manifested. If it's through death, then you have a result that's opposite to
Okay, so it is important in that setting. But I guess one question is, in terms of the rate, how does that compare? So you've got decreased divisions, increased death, but what are those relative rates? Like, is it half the amount of the proliferation rate? Is it comparable to the proliferation rate? Like, how different are they?
So what is the amount of disadvantage or advantage that we took? So we looked at different amounts. So for a simulation with 10 to the 3, n, when n is 10 to the 3, a very small size, it took the disadvantage of 17% to see this effect, where the result reverses. When the simulation, when the size was 10 to the 5, it was a smaller disadvantage threshold. So as you increase the colony size, this effect grows and it takes a smaller amount of disadvantage. But we look at all disadvantages from like 1% to 2-fold or 5-fold.
Sure. Okay, that helps. One last point then. How different would the results be if your mutant actually had a different death rate, division rate to your non-mutant?
Say again? If my
Basically, if you had differential fitness, both in terms of division and death.
Oh, great question. Working on it right now. So we, it's here. So this is the state space, and we explored two lines, horizontal line and the vertical line. You're asking about everything in between.
Yes.
Yeah. Somehow it'll be one or the other or in between, but it's a great question. Yeah.
Okay.
Yeah, I have a student working on it right now. And I don't know yet. Okay. Any more questions? So that's the problem with not seeing the audience. I just keep going and I don't take questions. Okay, sorry. So, okay, going to these different geometries. So we have a look at the cylinder with a flat side that goes one direction. Then we'll look at a sphere going in all directions in 3D, and finally exponential growth, right? And we want to look at the growth laws of mutants that are disadvantageous, neutral, and advantageous. So the easiest one is disadvantageous. So basically we want to know how many mutants can we expect to have in a colony of size n for all these different geometries. And I don't want to give you the exact formula because we have some formulas, but not others. We want to know kind of the order of magnitude. What is it defined by? So this grows as n. As n increases, it grows linearly with the colony size, because the disadvantageous mutants are produced and then it's mostly production. They don't really spread, okay? That's why we have these bubbles. But for neutral and advantageous mutants, we have all sorts of different laws. And for example, for neutral mutants, in a 2D flat front geometry, they grow as n squared. Compare this with the very well-known result of n log n in exponential growth, right? So it's faster. They grow faster. Neutral mutants grow faster in space. And this power actually decreases as you increase dimensionality. It's actually the fastest growth corresponds to the most restrictive spatial geometry, which is 1D. This is kind of like almost 1D because you only grow in one direction, right? So it's the fastest growth here. In 3D, it doesn't work like this. Oh, I'm sorry, for advantageous mutants, it's also always faster than in the mass action system, but you have different powers. So we derived those. There's a very simple derivation. You have to take like one integral, and you derive these laws, and we compare them numerically with simulations, and it always holds. We look at the number of mutants as a function of total population. And when you plot it on a log-log scale, you get straight lines. These are your scaling laws, the power laws. And these powers, the slope tells you the power. So and there are deviations from these formulas, which can be explained, but mostly it holds right. So this is something interesting that we found. And yes, we now work on, and by the way, what I mentioned before, that disadvantageous mutants grow differently depending on how they are disadvantaged. It's not even here, because they always grow proportionally to n. But there could be more of them or less of them than here, depending on the way that disadvantage is manifested. So there is no contradiction here. Okay, so I'll go on to my favorite, also another of my favorite topics, which is random environment. And I'm a little bit short on time, so I'll go, I may have to skip something, but so again, we'll talk about one mutation event, so just gain of function mutants. Suppose we have n cells in the box, and at each update we remove one at random and double one of the remaining ones, like this, right? So the question is, what will happen, right? And if you think about it, of course, they will eventually become all one color. Which color? It's any of the colors, right? The probability for them to become orange is 1 over n, where n is the number of cells. It's just simple symmetry. So in this process, eventually, all the cells will be clones of just one progenitor. Which one? Since they're all kind of the same, neutral, it's fair. So, and this gives us a very well-known law of genetics. The probability of fixation of a neutral mutant is equal to its initial frequency. So if we start from m neutral mutants, then the probability that everybody become like that mutant in the end, the probability of fixation is given by its initial frequency. Okay, so now higher or lower fixation probabilities indicate selection. So if my probability of fixation is more than this, it means that I'm advantageous, less than this, disadvantageous. And of course, the reason I talk about it is I want to show you how this law breaks if you introduce randomness in the environment. So we're going to go around a circle, for example. We can have a circular geometry or 2D, 3D geometry, I illustrated here. And I'm going to say that fitness of cells depends on where they are. Okay, so if I put a sunny spot means you're fit. So your fitness is 1 plus epsilon, and otherwise it's 1 minus epsilon, because say you're a plant that likes sun. Now let's consider, so this is the fitness of wild type. Now consider mutants. Let's suppose that mutants don't care about sun, but they need something else, like rain. So I randomly go around the circle and put raindrops in them. And whenever I see raindrops, if I'm a mutant, my fitness will be 1 plus epsilon. And if it's dry, it's 1 minus epsilon. Okay, so this is a random distribution of fitness, and the fitness of wild types and fitness of mutants are decided randomly from the same probability distribution. So it could happen, sorry, what's going on? Okay, so it could happen that by chance, we have 1 plus epsilon for wild types and 1 minus epsilon for all mutants, right? It can happen, unlikely, but it can happen. So this configuration completely favors wild types. So mutants are clearly disadvantageous here. It can happen with the same probability that the fitness configuration favors all the mutants and wild types are disadvantageous. Most of the time it's in between. So we are going to create this random fitness landscapes separately for wild type or mutants, or we can have them anti-correlated if they want, if it's easier. Okay, and so under specific realization of these, we are going to calculate the probability of mutant fixation. We fix the landscape, so we have good spots, bad spots, both for mutants and wild type. Put a mutant somewhere and calculate its probability to take over. Okay. So we calculated for a particular realization of fitness, then average over all realizations of fitness and obtain the mean probability of mutant fixation in a random environment. Okay, so the fitness of mutants and wild types comes from the same probability distribution. I'm not giving mutants any advantage or disadvantage. So we expect that on average, there is the probability of mutant fixation is going to be equal to the initial frequency, the law of genetics. So here's the case, n equals 3. It's an easy calculation. So a student, this is my favorite story, and I tell it for the sake of graduate students in the audience. So a student comes to me and says, I performed this calculation for the case of population size equals 3, and I found that mutants become fixated more often than they should. If they were advantageous, if they were neutral, they would fixate with probability 1/3, 1/n, starting with 1. But I find that it happens more often. So I say, no, that's not possible, right? I go home, I make my calculation, nothing. The student made a mistake. The probability of fixation is 1/3. So the amazing part is that the student goes home and does the calculation for n equals 4, and he has the guts to come back to me and say, you know, I did the calculation for n equals 4, and I found that mutants are advantageous. I was like, no way. And then it turns out that the student was correct. And so here I plot the amount of randomness, like basically by how much the advantageous and disadvantageous mutants differ from each other. And this is probability of fixation times 4. So if a mutant is neutral and everything is according to the laws of genetics, it's equal to 1 because the probability of fixation is 1/n, right? What he found is a little bit bigger. Look, it's like 0.2%, but it's there. There's no mistake. Then we do it for n equals 5, 6, 7, 10. This is probability of fixation times n. So it would be a horizontal line at 1 if there was no effect. Look at that. Okay, this is a 30% difference. We go to n equals 200, 800, 1,000, 15-fold difference. So the effect is not small, and the effect grows with randomness and system size. And it behaves the same way in spatial and non-spatial systems. So what we see is mutants without any evolutionary advantage behave as if they're selected for. And their probability of fixation is significantly larger than 1/n, which is predicted by the theory. And it only happens to mutants that are in minority. So you can start with 1 or 2 or less than half mutants. They feel advantage. They come to fixation more often than they should. If they're in minority, if they're in majority, no, it doesn't happen. Okay, so I wanted to step back and say, why do I even ever look at a circle, right? We started with a circle. So if we look at colonic crypts, which a lot of my work has been inspired by, these are folds in tissue, but this is a 2-dimensional cross-section. If you take this cross-section, you actually get a circle. So circular geometry is not out of this world, but of course, other geometries are even more important. And this effect holds for those too. And applications to cancer of this phenomenon are obvious, right? So cancers are characterized by very heterogeneous environments or microenvironments that include stroma, necrotic cells, blood vessels. The distribution of oxygen is non-uniform, as we all know, throughout the tumorous tissues. There are hypoxic regions, and the nutrients are distributed also in a complex fashion. And no two tumors are the same. Tumors have been compared to unhealed wounds. They produce all sorts of factors, inflammatory mediators. They have macrophages. They have stromal cells, all sorts of non-malignant cells that comprise the microenvironment. So basically, different spots in the tumorous tissues are different. So the fitness of cells, which always is in most calculations is taken to be a constant, is not constant. And by fitness, what do I mean by fitness, right? It's simple. Fitness is how well a cell grows and divides, right? And how little it dies. Like it's this composite quantity that reflects the ability of cells to reproduce, and that includes reproduction rate and death rate. So usually, like in my previous calculation that I showed today, all the wild types had the same division and death rate. All the mutants had the same, right? This is not true. It's really a random quantity and depends on where you are, if you want to truly consider a spatial model. So evolutionary models in random environments actually behave differently compared to any theory that have been done so far. And so I'll give you an intuitive explanation of why minorities, neutral mutants that are in minority may feel a selective pressure to expand more than it should, right? So if you perform a computer simulation and look at the mean fitness of mutants that start from a minority, you can see that as a function of time, it grows. Somehow as mutants spread through this spatial system, they gain fitness. And here's an explanation of why it's possible if you're a minority. Let's have a particular configuration where orange is a good spot. And I start with a small number of mutants, less than half, okay? And on average, for an average configuration, there are as many good spots as there are bad spots, right? If I do it many times, on average, it has to balance. Otherwise, mutants are on average disadvantaged. I don't want that to happen. So on average, there are as many good spots as there are bad spots. So as a minority, I can spread out, expand, and actually only put my progeny on good spots. In this process, the average fitness of mutants has increased, right? Now let's start with a majority. This is a wild type that was originally in the majority. And let's suppose that blue is good for a wild type, or actually orange here. So if I'm in a majority, it's very difficult for me to redistribute, to take only the good spots, because there are too many of us. So the effective process where I gain advantage, where I increase my fitness, can only work if I start from a minority. Okay, so that explains kind of in the hand-wavy terms why minority mutants have a selective pressure. And there is a mathematical explanation also, which we'll provide. So under random selection, evolution moves in counterintuitive ways, especially if you are in random environments. Minority mutants whose parameters come from the same probability distribution may become selected for. So I want to conclude this by saying that there are a few things that are more fascinating than mathematics of evolution. I want to thank my group and my collaborators and the funding agencies. And these are a couple of books that we wrote a few years ago that contain some of this stuff. But as always, the most interesting stuff is not in the books, it's work in progress. Thank you.
Thanks, Natalia, that was great. And as Heiko pointed out, I should know we're actually using your book with Dominic as part of our course in mathematical oncology. So thank you for that.
Thank you. That's very nice to hear.
So we have a bit of time for questions. You didn't have to rush through that because we deliberately planned to have extra time. So if we have any questions from the audience, please jump in and speak.
Yeah, hi, it's Heiko again, if I may. In the last part, Natalia, that you discussed the red and blue circle with the minority wild type cells having access to the good environments. If these mutant cells are in a minority, aren't the good spots in the environment occupied by the wild types? How could the minority occupy these good spots so beautifully?
Oh, so the process that we consider is a Moran process where all the spots are filled. There are two types of process, birth, death, or death, birth. Let's consider one of them, death, birth. You kill one cell fairly and then reproduce one of its neighbors in proportion to its fitness. So if a cell has two neighbors, you compare their fitness with each other, and the one that's more fit is more likely to take the spot. So as time goes by, starting with a couple of mutants or one mutant, they spread, right? or they die out. Okay,
Right.
it goes differently. So, but their spatial locations change, they redistribute, right? So as you watch a simulation, they occupy different spots from where they started from. And on average, this is a result of very many simulations. And again, it doesn't show any variance, but this is the mean fitness over many, many runs of a simulation, which measures the average fitness of mutants. We always start here with one mutant, and sometimes they die out, sometimes they expand, and at every time step, we measure their average fitness. And on average, it grows because they are successful in spreading. So, and the reason is that if you happen to occupy a good spot, you are more likely to divide, and then your progeny again divides, and eventually you on average you occupy more good spots than bad spots. That's all.
Do you have in this kind of plot a survivor bias fallacy in here? Because every time your mutant dies out, its division rate is no longer recorded. So, right, so in the end, you only record the division rate that leads into this plot here for the cells that survived, correct?
It's only the division rate of the mutants that exist at a given time point.
Right.
Yes.
So it's a bit of a survivor bias, isn't it? Because you ignore all the ones where the mutant actually was unsuccessful.
Yes, but that's a good point. But if you now plot, which I don't hear, but it's in the paper, if you plot the mean fitness of wild types, it goes down on average because they're a majority. So we call mutants a minority type and wild type the majority type initially. So here we start with m equals 3 out of 9 cells. So it's a minority at the beginning, and the line goes up. If I start with m equals 6 or 5 or 8, the line would go down. So it's the same process, but it works differently depending on whether you start as a minority or a majority.
Thank you.
Mm-hmm.
I think Artem's got his hand up. Artem, do you want to ask your question?
Yeah. Nice talk, Natalia.
Thank you.
I'm interested if you guys analyze the fixation times for the random process.
Yes.
Because usually when you have the selection towards the minority, you end up going from sort of a quadratic fixation time to like an exponentially slow one. Does this happen in your case, or is it what's the scaling like for the fixation times?
Let me remember. We have a whole paper devoted to this. Let me try to remember the results. So there were some interesting and counterintuitive results related to random environments depending on the network. So let me remember. So if you have a spatial network, for example, a 1D circle, then having randomness delays fixation. So what we measured was the conditional expectation of fixation. So conditioned on the mutant reaching fixation, we measure the average fixation time. That's the standard measure. And we compare that with the same system without randomness. And so if you are on a circle or in any spatial system, then having randomness makes fixation longer. But if you're in a well-mixed system, having randomness accelerates fixation. And there is an intuitive explanation of that. If you're in a strictly spatial system, having a bad spot next to you can delay you forever. But if you are in a mass action system, being able to jump to a good spot accelerates you, and that works. It contributes differently to the dynamics. So we have these opposite results for the type of the network or spatial configuration of mutants. So that's the most striking result I remember, but we have actually determined all sorts of things, like how, so your fitness can be defined by your random division rate, random death rate, and both fixation probabilities and fixation times are affected differently depending on what defines your fitness. So there's a bunch of results, some of which we understand.
Okay, thank you. I'll look into those. Thank you.
Okay, I'm not sure if you have any other questions. No, I'm not hearing any. But very interesting, Natalia. We've also been looking at some aspects of this, not quite with the same rigor as you. I do really like this idea of kind of stepping back and building the simple ODE model to sort of help you understand what these stochastic simulations are showing you. It's a really nice approach. We've been particularly interested in really how spatial constraints of different sizes and different dimensions potentially can impact, you know, basically, in some sense, different the coexistence of different driver populations. And then, of course, ultimately that could lead to something like fixation. We were seeing much faster sweeping in smaller domains, and then, of course, more coexistence in larger domains. And then if you imagine now that in a kind of architecture that would be something like a ductile tree, there could be very different domains that a population is seeing. And so a long way of saying, my question to you is, how different do you think this would be if this was a metastasis that seeds in a tissue where it's literally competing with a normal tissue versus something that's, you know, basically growing into empty space the way you've modeled it? How impactful would competition from non-tumor be potentially?
So that's a good question. So when we look at this expanding spot that grows into empty space,
yeah,
that actually is the dynamics of the mostly the wild type. So if a mutant appears in the midst of this colony, it no longer grows into an empty space, but it invades the surrounding tissue comprised of wild type cells. But I hear what you're saying. We need to somehow put this in the context of a microenvironment. So we are starting to look into this in terms of stroma, which restricts the geometry. And I think it's very complex, actually. Because not only do you not grow into empty space, there are also factors that are produced by the stroma that affect your growth. And depending on how you handle those, everything can change. So you noted my tendency to simplify to the bone. So this is the next step, and we are trying to look into this. What's the kind of the gist? What can we learn that we can actually understand from this? So we started by looking at signals that cells send to each other and how they affect each other. And that's so complex, even in the absence of stroma. We're doing it right now, and it's mind-blowing how the spatial spread of intercellular signals can affect the dynamics. So yeah, there's lots to do there.
Indeed. And of course, you know, we all have to start somewhere, but it's cool to hear that you're adding and thinking about adding these other components that are taking it towards some sense more realism. But of course, we have to be careful about, do we make it so complex we can't understand it, right? So I think the route that you're doing makes a lot of sense. We're definitely interested in the role of stroma too. And of course, stroma can be this, you know, can play different cards. It can be promoting, it can be inhibitory, and, you know, and it can be transformed by the tumor itself. So there's all those complicating dialogues going on, and you're just trying to understand one aspect of that dialogue. It makes it hard. But I think we have to try and tackle these questions, even if they're tough, right? We have to try.
Absolutely. Working day and night on this.
Great. Well, if there's no other questions, thanks, Natalia, for a great presentation. It was lovely seeing you again.
Thank you very much. Very nice to see you too.
Okay. Thanks. Thanks,
Bye.
everyone.